Okay, guys, so let's start talking about the physiology of the male reproductive system. And so when we're talking about the male reproductive system, we have to start talking about, when we're talking about the physiology, it's going to be enacted through the sexual response. And there are two parts of the male sexual response. There's both an erection and ejaculation. So, the erection. An erection is just an enlargement and stiffening of the penis. And basically what happens is that arterioles are normally constricted. And in sexual excitement, it's going to cause the central nervous system activation of parasympathetic nerves. And they're going to release nitric oxide. And basically, nitric oxide release causes relaxation of the local vascular. When the smooth muscle can relax, the arterioles will dilate. And basically, when this happens, the corpora cavernosa expands and it retards the venous drainage. And because it retards this drainage, basically, that the blood can't leave, it's going to lead to the engorgement of erectile tissue with blood. And this is what's going to cause the penis to stiffen and enlarge. The corpus spongiosum is important because it helps to keep the urethra open so that semen can leave, right? Now, erectile dysfunction can be a problem, and basically this happens when the parasympathetic nerve of the penis releases too little nitric oxide. And this can be because of alcohol, it can be because of drugs, hormones. There could be either blood vessel or nerve damage. And it can be a problem, and I'm going to put it up here so that you can see it on the side. It can be an incompetent venous valve that basically fails to retain blood in the penis. Now, there have been drugs like Viagra and Cialis that potentially exist to help to basically fix this problem. Now, with erection, overall, if you don't have this erectile dysfunction problem, that basically an erection is initiated by sexual stimuli, so such as someone touching or mechanical stimulation of the penis, things as much as erotic sights, sounds, and smell. And they can be induced or inhibited by emotion or higher mental activity. So basically keep that in mind. And there are basically longitudinal and circular collagen fibers that are around the penis that are preventing it from kinking or buckling of an erect penis. Now, the ejaculatory process of male response is when you have the propulsion of semen from the male duct system, and we talked about the duct system itself. And this goes from a sympathetic spinal reflex So here the bladder sphincter muscle constricts, preventing urine from leaving, right? Because there's never that mix. And basically the duct and accessory glands are going to contract and empty their contents into the urethra. And you have the bulbospongianosus muscle that undergoes a rapid series of contractions that's going to cause the expulsion of the semen, right? So this is the muscle there that actually allows for it to leave. And it's closely to 11 miles per hour, right? And so the ejaculatory event itself is called climax or orgasm. And I have already talked about this, so I'm going to move past that. So let's go on really quick. We talked about inside the semen, there are sperm. But how do we actually produce the sperm? How do they get made? The process is called spermatogenesis. And spermatogenesis is the actual formation of brain, the sperm, I mean. And you can think Genesis as if you think of beginning, and you see sperm in that word. Now, they're also called spermazoas, so don't get confused on that. And this production actually happens in the seminiferous tubules, and it takes between 64 and 72 days. Now, sperm are interesting because they only incorporate one set or half the set of chromosomes that everybody has. So most body cells have 46 total chromosomes. There's 2 sets of 23 pairs of chromosomes, right? Gametes only have 1 pair. So this up here is what's known as a diploid cell. And basically these gametes or sex cells are known as haploid. And you can tell the difference. A haploid cell is 1N and a diploid cell is 2N. And so this whole process of spermatogenesis, that's specifically for the production of sperm, uses a process known as meiosis. And I like to think of meiosis as making little me's. And so basically what they do is they take these diploid cells, regular body cells, and they cut them in half. Right? So you go from 2N to 1N. And this is going to help induce genetic diversity, as all daughter cells, or those are your offspring cells, are genetically different from the original cell. So let's talk about meiosis, and this is going to be a video that helps to introduce it.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis II, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
So that kind of gave you an overview on what we're going to talk about. So mitosis we hit on in A&P 1 towards the beginning of the semester. And mitosis is a way to basically allow cells to reproduce when we're growing or to help replace when they become old or damaged. And so basically what happens is you have a mother cell or the initial cell, and basically what happens is they duplicate the number of chromosomes and they divide once. And basically from there you get the exact same cell. That should make sense whenever you're trying to replace skin cells or some other type of cell like that. Now, how that differs from meiosis is that you still have that duplication process at the beginning. That happens during the S phase of interphase. And then you actually have the process of meiosis happening, where essentially you are going to separate all the homologous chromosomes are going to separate, right? Here, you can see that you have the homologous chromosomes together, and basically they separate into sister chromatids. They move apart. Where here, the very first division is just the homologous chromosomes. So when we come to, and that's what's known as mitosis 1, or meiosis. I'm sorry about that. In the second part, you're going to get the division of the sister chromatids. And you can see from here that each one is by themselves. And this is what essentially is going to pick up the gametes. And at this point, we have the haploids, or N. So this is going to have 2 divisions, where this one has 1 division, right? This is what's known as meiosis 2. So this is the main difference between the mitosis and meiosis, is that you start with a 2N cell. In mitosis, you end with a 2N. In meiosis, you end with a single cell. And you have 2 divisions in meiosis. We also have something known as crossing over that happens in meiosis. That doesn't happen in mitosis because the homologous chromosomes, the set that looks like your mom and the set that looks like your dad, that look similar, they never come together in mitosis. So let me really quick show you and do a comparison between meiosis and mitosis. Since you should already know mitosis, if you don't, then you should go back and review it really quick, because the stages or what happens a lot have similarities between that and mitosis and meiosis.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
So hopefully that helped you to kind of understand the difference between the two. So let's actually talk now what it is. So meiosis 1, this is going to be the very first separation. So you're going to have within meiosis 1, and you always have to put the 1 behind it. That's telling everybody that you're in the first division. The first part of meiosis is actually prophase 1. Now there is a stage right before that's called interphase. Interphase is a preparatory stage. It's not part of meiosis. And basically it has 3 different stages. It has gap 1, the S phase, and gap 2. And basically the G phase, what's happening there, both of them, is that you're having growth of the cell. This is where the cell becomes double the size. You're getting more organelles. All of those activities are happening in G1 and G2. During synthesis, this is where DNA replication happens. And we talked about DNA replication last semester. I'm not getting really big into this because this was already covered in part 1 of this course. And again, if you don't remember this, then you need to go back and search through that material and just get brushed up on this. And so once we get done with that interphase, we go into meiosis itself and prophase 1. Now, prophase 1 here, just like in mitosis, all those same things are going to happen. You're going to get the nuclear envelope is going to start disintegrating. You're going to have the spindle or the centrioles go to opposite sides of the pole. You're going to have the chromosomes start to condense. So all of those things that you remember from mitosis is going to happen in for prophase are going to happen in prophase here as well. The difference here, a big difference, is that you get the homologous chromosomes that are going to be pairing. So the chromosomes that you have from your mom that have the same, like chromosome 1, is going to pair with chromosome basically 1 that your dad gave you. And because they're so close to each other, they're attached, they're going to have what's called crossing over. And this is where basically genetic recombination is important. And so here you can see it. Here are the homologous chromosomes. They look the same. The red is from mom, blue is from dad. And you can see on each one of those you have sister chromatids coming together. So they're going to form what's called a synaptic complex here, right, on the ends. And this is basically where the synapse begins. These are what are known as triads or bivalents, right? Triads because there's 4, bivalent because there's 2 of these coming together. Now at this point, crossing over can occur, and you can see that's when you take a bit of genetic material from mom and a bit from dad and you flip them together, and that's what's known as a chiasma. You can see this here. And so here's a picture of what a chromosome looks like. And you can remember the kinetochores here. That's the area that basically is going to be on the outside of the centrosome in the middle. That's going to connect to the microtubules when they move those around. Some of this I'm assuming you know because it's been taught in part 1. And so from here, you can see a chiasmata here and here. And so because this can happen, you can see the arrangements of these sister chromatids here can be quite different between offspring. And this is the reason why not every single offspring looks exactly the same. Because if you look, this is the original that you have for your dad. This over here is the original from your mom, right? If you look here and here, these are completely new, right? You have all the dads with a section of mom on there. So imagine if you get the arrangement of different possibilities you can have with crossing over, and then when you actually go to line these up in the middle, that the way they line up here is independent. Each chromosome pair that comes to the middle is independent of the other. It's called independent assortment. So they can line up all differently in the middle. So what kind of chromatids that you have in each gamete can be drastically different. So this is what meiosis looks like. So here's interphase 1. Oops. And you can see nothing big here, but prophase 1, this is mid, and late. You can see that the nuclear envelope is gone, and you can see these homologous chromosomes are pairing up together, right? You're going to have the centrioles going to opposite sides. You can see them here, right? And you're going to start getting the development of the spindle fibers happening. That's what attaches to the chromosomes itself. And so from here, metaphase, anaphase 1, the exact same thing happens as in mitosis. And so let's look at here, metaphase 1. You can see all this homologous chromosomes are meeting in the middle, down the midplate. This is the part where the chromosomes are actually the most condensed as well. Here, the homologous chromosomes are separating. So you get one set here and one set here, right? So they're pulling apart. You can think M, middle, A from away. And basically, telophase is the opposite of prophase, is what. So except for the the crossing over and all that, all the other things that are happening in prophase, basically the opposite is happening here. So the nuclear envelope's coming back. The chromosomes are uncondensing. You have the centrioles are now together. The spindle fibers are gone. So everything that's happening at the beginning of prophase or through prophase, the opposite is happening in telophase. And if you notice now, we have two cells. So number one, for the most part, if you see one cell there, you can think you're in meiosis 1, all the way till you get to the very end of telophase. Now within telophase, you have what's known as cytokinesis, and this is the separation of the cytoplasm. Meiosis 2, this is when you're taking the sister chromatids that are together and separating them out, right? Now each form of chromatid is now going to be their own chromosome. So if you see, all the same things happen in prophase as before, and you can see that they're starting to condense. And this is prophase 2, and of course you can see two cells. They're meeting in the middle, and again, you see two cells. There's just a separation in them. The middle of the two, but they kind of go together. Anaphase 1, M for middle, A for away. Here you're having the sister chromatids separate into opposite sides. So before you had two chromosomes together and one would go this way and one would go this way. Now these guys are separating and they form chromosomes on the opposite poles. And then at the end of this, for telophase 2, you're going to see, oops, I'm sorry guys, 4 cells here, right? Of course, cytokinesis is happening here. And these, you're going to go from 2N to now they are haploid 1N cells, right? And the same thing happens here for telophase. So I'm going to show you a video that kind of goes back over all of it and kind of reviews both mitosis for you and meiosis. Then we're going to talk about independent assortment, kind of review, and then go into how this is happening in the different stages of the cell in spermatogenesis, and actually show you where you have these spermatogenic cells and basically how they go from spermatogonia and through the different stages till you become sperm. whole process all the way through for both. Now, when we're going through the process of meiosis, there is this idea that's called independent assortment. And what it means is you have each set of homologous chromosomes lined up in the middle, right? And the orientation that they line up is independent of the other. So you just see how this one over here has both green on one side and blue on the other? Well, this guy easily could be the opposite. And if that happens, you can see how the combination of daughter cells changes. And then imagine this 23 times. So it does allow for this genetic diversity in addition to the crossing over. And so basically this is just the whole set going all the way through. And I'm not going to spend much time because I've shown a couple videos now and I've talked, but you can look in your book and get a really good idea of the separation and how meiosis and 1 and 2 differ from each other. Now, when we're talking about spermatogenesis itself, this is a process that happens in the seminiferous tubules. And so this is where the spermatogenic cells are going to give rise to sperm, and it takes about 64 to 72 days. And there are these basically overall steps to spermatogenesis. The first one is mitosis of spermatogonia. These are the stem cells, and these are going to be what forms two spermatocytes. At this point, the spermatocytes will undergo meiosis, and they will form secondary spermatocytes, which then will turn into spermatids. And the spermogenesis is the process of spermatids, which are not active sperm, becoming the spermatids coming sperm, right? Don't get the spermogenesis confused with spermatogenesis, which is the entire process of it. So let's look at a picture and talk about how it works. So let's look over here. And basically this testes, particularly what's happening in the seminiferous tubules. So you'll notice here, right? That is what's called the basal lamina. And attached to that are these things called spermatogonia. These spermatogonia are the stem cells. And basically, right here is where mitosis happens. So you can see these line up with each other. So right now, this guy right here is this guy right here. So they're going to undergo mitosis. And they are going to divide basically into daughter cell A or daughter cell B. Now, this whole process or spermatogenesis essentially begins at puberty. It doesn't happen early on in life. And basically, this starts with the spermatogonia, right? Again, these are stem cells. And so what happens is when you become daughter cell A here, right, you remain onto the basal lamina and you are going to end up being a germ line. This is how they keep constantly being able to have those cells, because the cells will get old. And so they divide. One stays at home by that basal lamina to continue the process. The other is going to go down, and this is the B cell line, right? And this is going to move towards the lumen, and this is what's going to develop into what we know as the primary spermatocyte, right? So at this point, and you can see here, this is A, this is B, and this is a primary spermatocyte, right? If you notice right here, you'll see there's a tight junction during that process. Do you see that guy right there? We're going to talk a little bit later about these tight junctions, but just to let you know, they form blood testis barriers. As I was telling you earlier, sperm's not formed until puberty, right? So it's actually absent during the immune system development. So this guy right here is going to prevent sperm antigens from escaping into the blood and causing activation of the immune system. So if sperm aren't there when the immune system's developing, the body won't be able to recognize it as a self cell. So it would essentially be attacked. So to avoid it from being attacked, there are these tight junction forms there to basically prevent it from escaping into the blood. Now, so that's mitosis, right? As you can see, we originally had the 2N cells here, right? And we're still at 2N. And at this point, once we get the primary spermatocyte, that guy is going to undergo meiosis. So here we get the primary spermatocyte, which is 2N, and he's going to undergo meiosis 1. And that is going to basically form 2 secondary spermatocytes. So you see here, meiosis 1, so you're going from N, 2N to N, and these are the secondary. And during that time, right, you're going to also, right after that, you're going to get your second division, right? So this guy is going to divide here and here. So each of these secondary spermatocytes are rapidly undergoing meiosis 2 to become 2 spermatids. And you can see here, these are early spermatids, right? And mitosis 2, or meiosis, I'm sorry. So this is where we get spermatocytes changing to spermatids, and this is meiosis. So this top part, mitosis, the second part here is meiosis, and the last part we're going to look at is the spermatogenesis, which is taking these spermatids and turning them into sperm. And so basically what's going to happen here, they all have the correct number, N, or the haploid number, that they need for fertilization. These guys, though, aren't able to move. So they're unable to swim, but the pressure of the testicular fluid pushes the immobile sperm into the epididymis, where they can gain motility and fertilization power, right? But during this spermatogenesis, basically it's a streamlined process where the spermatids are going to elongate. You see how they go from being just circles and then they become elongated. They're going to basically lose cytoplasm. They're going to form tails. And they're going to go into these 3 different regions, right? So this is the last part, and you can see it over here as well. So this is this process of spermatogenesis. Now let's look really quick at spermiogenesis so that we can just take a quicker look at it. This takes approximately 24 days to happen. And as you can see, this is the earliest spermatid, right? And essentially, you can see over here, the centrioles disappear, and you can start seeing the microtubules helping to form the flagellum. And basically, these mitochondria will line up towards that middle part. It'll form the chromosome here, and that is one part. This is part of the head region, right? And this is what's going to have the genetic material. That's going to basically have the nucleus. And this chromosome here, it basically has the hydrolytic enzymes. Those hydrolytic enzymes, if you remember, they help the sperm to penetrate the egg. Excess cytoplasm is leaving there, and you can see it's starting to form in that middle area, the midpiece. This is the metabolic region. So that's going to produce all the ATP. As you can see, the mitochondria are lined up there. And the last part is this tail region. And that has a flagellum, and so it's for locomotion or movement. So this is the process of making functional motile sperm. Now there are supportive cells there, and they're called Sertoli cells, right? And they have several different functions. So they extend through the walls of the tubules. And basically they surround developing cells. They provide nutrients. And they help signal to dividing cells. Essentially the cells that will make sperm. They help to move cells along to the lumen. They secrete testicular fluid into the lumen that help with sperm transport. They help to phagicize, faulty germ cells, and all the excess cytoplasm. Don't need stuff, so they get rid of it. And they produce chemical mediators. These guys are going to regulate spermatogenesis. And again, they contain tight junctions that help divide tubules into two compartments. These tight junctions, again, prevent sperm antigens from escaping into the blood and causing activation of the immune system. And that's because sperm is not formed until puberty. So it's absent during the development of the immune system. And so the body can't recognize sperm cells as being part of its own body cells, so it will attack it. So it's a way to separate it so that doesn't happen. You have the basal compartment. This is an area where the spermatogonia and the primary spermocytes are located. And the abluminal compartment is the area where metabolically active cells and tubulins are located. And so there's a way that we regulate the reproductive system, and that's through the hypothalamic pituitary gonadal, or HPG axis. And this is for the production of gametes and sex hormones, how they're regulated by the sequence of hormonal events involving the hypothalamus, the anterior pituitary, and the testes, right? And it involves these set of different hormones. You have the gonadotropin releasing hormone, you have the follicle stimulating hormone, you have the luteinizing hormone, you have testosterone, inhibin. So all of these work together. Now I kept these here so that you can read through them, but I'm going through and we're just going to actually look at the picture as we go through here. So the very first thing that happens is that the hypothalamus is going to release GnRH, which is the gonadotropin releasing hormone. And so this is going to go from the hypothalamus to the anterior pituitary gland, which essentially then going to release follicle stimulating hormone and luteinizing hormone. The follicle stimulating hormone is going to go down and it's going to stimulate spermatogenic cells to start producing sperm. And it does this indirectly by stimulating those cystinocytes to release androgen binding protein, otherwise known as ABP. And so ABP keeps concentrations of testosterone high near spermatogenic cells, promoting spermatogenesis, and that's how it works. Now the luteinizing hormone here is going to bind to interstitial endocrine cells. And that's going to prod them to secrete testosterone. Rising testosterone levels, right, are basically going to stimulate sex organ maturation, develop and maintain of secondary sexual characteristics and libido. All these things we're going to talk about here in a second. Now, testosterone or rising levels of testosterone is going to lead to feedback, though, of the hypothalamus. And once those are high enough, right, so you're going to see that it increases spermatogenesis. It helps with primary and secondary sex characteristics, primary being the actual sperm, secondary being all the things that you see on the outside. But then high testosterone levels will lead to negative feedback. So testosterone will come back to the hypothalamus and inhibit GnRH production. And inhibit basically the pituitary system from releasing basically GnRH, and that will stop the FSH and LH, or follicle stimulating hormone and luteinizing hormone. Now there's also something known as inhibin, which basically is released by cystinocytes when sperm count is high. And basically this inhibits this and this. So this is how basically sperm or spermatogenesis is regulated. The amount of testosterone in sperm produced by testes basically reflects the balance among the three interacting sets of hormones, specifically the gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone. The balance takes about 3 years to achieve, after which testosterone and sperm production are fairly stable throughout life. Without the gonadotropin releasing hormone and gonadotropins, testes will atrophy and sperm and testosterone production ceases. Before birth, male infants have testosterone levels about 2/3 of adult, but soon after birth, the levels will recede and remain low throughout childhood until puberty. And so this is basically showing the basically levels of testosterone. You can see at fertilization they're high, but during those basically from about 3 to about 5 months that it peaks, and then it slowly comes down till birth. It peaks really quick at the beginning, and basically, and then once you hit puberty until adulthood, it gets really, really high until about the age of 60, where it starts falling again. Basically, testosterone comes from cholesterol. It looks very, very similar to it. So basically, cholesterol, when you look at it, it ends up helping to form both estrogen, or cholesterol is used to form both testosterone and estrogen, obviously, the two main sex hormones that are in male and female. It can be converted to what's called DHT, or dihydrotestosterone, in prostate, and estradiol in some brain neurons. This helps to promote spermatogenesis and targets all accessory organs. It can have multiple anabolic effects throughout the body. Deficits can lead to atrophy of accessory organs, semen volume declines, erection and ejaculatory can be impaired, and basically treatment is used for testosterone replacement. Okay, so when we're talking about testosterone and its effects, the primary sexual organ that we're looking at is essentially the gametes. But testosterone can also affect what's known as male secondary sexual characteristics. And these features induced in non-productive organs by male sex hormones, mainly testosterone. And basically, think about it. It's an easy tell-all way for the body to show an outward way of what type of gamete you have. Imagine if everybody looked like a male or a female, how hard it would be to determine which person you would need to pair up to reproduce. You would be going to basically all individual people and going, hey, do you have this? I have this. And they're like, well, no, I can't reproduce you. Do you have this? I have this. Oh, no. So these are ways for which we can basically physically see if someone has the male primary sexual characteristics. And I'm going to have to, here, thank goodness. So let's see what those are really quick. So these basically features induced in non-reproductive organs by male sex, so mainly testosterone. So we have the appearance of basically pubic, armpit, and facial hair. We have an enhanced growth of chest hair. Deepening of the voice. thickening of skin, and it becomes oily. Bones grow, and they become more dense. Skeletal muscles increase in size and mass. You have a boost of basal metabolic rate, and this is basically, I'm going to add a little 8 in here. It's the basis of sex drive or libido in males. So with that, it finishes the end of our male physiology section, and next will be female.